You know that feeling when you settle in with a really nice glass of wine? You swirl it, you notice the color, you take in that amazing aroma. But what if I told you the most important artists behind that wine aren't even on the label? Well, we're about to meet them. Because that beautiful glass of wine, it's basically the end result of a microscopic ballet performed by literally trillions of living organisms. All those complex flavors and amazing smells, they all come from the hard work of yeast and bacteria, which take simple grape juice and turn it into something truly special. So that kind of brings us to the big question, right? If you're a winemaker, how on earth do you manage this invisible world to make sure you get a perfect result every single time? What's the secret? Well, the secret is all about managing this massive, invisible population. There's a whole field for it called enological microbiology, and it's really just the science of understanding and guiding all those tiny yeasts and bacteria that are the true architects of wine. Okay, so if you're a winemaker, where do you even begin with this invisible world? You start with the most basic tool in the lab, the microscope. It's your very first peek into the universe that's just teeming with life inside every single drop. And just by putting one drop on a slide, you get this incredibly important snapshot. You can ask, okay, are there any microorganisms in here at all? Can I tell the bigger yeast cells apart from the tiny little bacteria? Does everything look healthy and diverse, or does something seem off? It's basically your first line of defense. But here's the catch. There's a huge blind spot when you're just looking. The microscope can't tell you the one thing you really need to know. Which of these little guys are actually alive and working, and which aren't? Which, of course, leads to the next big question. We can see them, sure, but how many are actually in there? To make great wine, you have to manage the size of the crowd, so you've got to do a head count. And this is the tool you use to do it. It's called a hemocytometer, or a counting chamber. Now, it might look like a regular microscope slide, but if you look closer, it's etched with this incredibly precise microscopic grid. And that grid, that's the whole secret. It lets a winemaker count the cells in a very specific, known volume, which means they can then calculate the total population. Now, the process itself is super meticulous. You start with a good representative sample. You might have to dilute it down, then you carefully put it into the chamber, you count up the cells you see in the grid, and then, with a little bit of math, you get your overall cell density. And that term, cell density, that's a really key metric for us. It's the total number of cells, remember, both living and dead, in the wine. So you can think of it as just the total size of the crowd we're trying to manage. But just knowing the total number of cells isn't enough. I mean, think about it like this. Imagine you're managing a factory. Knowing how many employees are physically in the building is one thing, but knowing how many are actually clocked in and doing their job, that's the number that really matters. And it's the exact same thing in wine. So this brings us to a really crucial difference, the total count versus the viable count. That total count we got from the hemocytometer, that includes every single cell, living or not. But the viable count, that tells us who's actually on the clock. It's the living, working population that is actively turning sugar into alcohol. That's the number that really tells you what's going on. So how do we figure that out? Well, winemakers have a couple of classic methods they use. The first is a special dye called methylene blue, and the second is a technique called plate counting. Methylene blue staining is actually a really clever and simple trick. You see, living yeast cells have these enzymes that can break down the blue dye, so they stay clear. But dead cells can't do that, so they soak up the stain and turn bright blue. By counting the blue ones versus the clear ones, you can get a pretty quick idea of how many of your cells are viable. Now, for an even more accurate count, you've got to use a technique called plating. The thing is, a single drop of fermenting wine has millions and millions of cells. It's just an impossible crowd to count. So, first, you have to thin them out. This process is called serial dilution, and it's a brilliant way to take one part of your sample, mix it with 9 parts of a sterile liquid, and just repeat that until the population is way more manageable. So what do you do with that diluted sample? Well, you take a tiny amount of it and spread it onto a petri dish that's full of a nutrient-rich gel. It's basically a perfect buffet for any living microbe that lands on it. And then, after a few days, something really cool happens. Every single living, viable cell that landed on that plate, it multiplies over and over again until it forms a visible dot, which we call a colony. So by just counting those colonies, you get a super accurate measurement of the living working cells from your original sample. This is really the gold standard for getting a viable count. Okay, so we've seen them, we've counted them, and we know who's alive and who's not. But there is one final, and maybe most important question to ask. Who exactly are they? Identifying the key players is the last critical piece of this whole puzzle. And this is where things get really high stakes, because in this microscopic world, you've got heroes and you've got villains. Some yeast strains will give you these beautiful, floral, fruity notes, but then certain bacteria can come in and produce these awful off flavors that can completely ruin an entire tank of wine. So yeah, identification is everything. Now, while there are traditional ways to do this, a lot of modern wineries are turning to this whole suite of high-tech, rapid methods to get faster, more precise answers. These are seriously powerful tools that can identify not just the species, but the specific strain of a microbe. Let's just focus on maybe the most famous one of these, PCR. I'm sure you've heard of it. Honestly, the best way to think about it is as DNA fingerprinting, but for microbes. The technique can find one unique little piece of a microbe's DNA and then make millions of copies of it. This gives winemakers a definitive, super fast ID, which is just an incredible tool for quality control. And this all comes back to a really simple but profound truth, one that's captured perfectly here. It's the metabolic activities of these microorganisms that influence the sensory qualities of the final product. In other words, they don't just make the alcohol in wine, they are the wine's character. They are the unseen artists. So the next time you pour yourself a glass of wine, just take a second and remember what you can't see. Look past the color and the aroma and think about the journey. Think about the billions of tiny, living architects that performed this incredibly complex ballet just to create that experience for you. Because you're not just tasting grapes, you're tasting the art of an entire microbial world.